nowadays, more than 5 years ago, talk about gene therapies, that pharmaceutical companies are entering the field of gene therapies, that these are expensive medicines. Thank God we don't have to deal with reimbursement. But also you may have heard on the treatment success of some of these therapies, like for example the CAR T cells. So these treatments have a 10-year tradition, more than 10-year tradition in the European Union, and the ATMP regulation, the specific next special law came into force about 10 years ago, and this law says that advanced therapies are medicines, that they are authorized via the centralized procedure. That means that the European Medicines Agency is responsible for the coordination process of these medicines. It also stipulates that these products are assessed quality, safety, efficacy by a specialized committee, and that's the Committee for Advanced Therapies that was founded in January 2009. So we were celebrating the 10 years existence of the CAT. And in the meantime, we have GMP and GCP guidelines that are adapted to ATMPs, and we also have a specific post-authorization vigilance and risk management. And if a product is either a gene therapy or a somatic cell therapy or a tissue engineering product, these three classes are summarized under the umbrella of advanced therapies, and then these products are assessed by the Committee for Advanced Therapies and not by the other committee, which is the CHMP. And here you can see the 14 products that we have authorized since 2009, and you see in green those that we call tissue engineering products with 3 cartilage-containing, chondrocyte-containing products for repair of knee cartilage. Then you see in blue products that we call somatic cell therapies, and in red those that are classified as gene therapies. And you can see that there is a broad range of indications, from orthopedic indications through ophthalmology, through hematology, oncology, to the very rare diseases like the inherited retinal dystrophy disease, where we've authorized Luxturna last year. And here you can see how we work. So you see here the CAT, a committee where all the 28 member states are represented, and from these Committee for Advanced Therapies we select the rapporteurs, and I'm very happy to have one of the CAT rapporteurs here in the room. So we assess quality, safety, efficacy, and then in that assessment report is then going towards a draft opinion passed on to the CHMP, the Committee for Medicinal Products for Human Use. And so that's the scientific assessment, and then pass the assessment, the evaluation on to the European Commission, who is then responsible for the authorization for placing the ATMP on the market. And you can see here an overview of all of the other tasks, the main task being the evaluation of a marketing authorization, but there are also many other tasks we are involved in the EMA scientific advices for ATMPs. We have, in the meantime, in these 10 years, published a lot of guidelines for gene and cell therapies, and you can see here full house is with members and alternates, 68 people, the 28 from all the member states. Norway, Iceland is presently not represented, but we also have clinician and patient representatives. And in our committee, this is a picture from last year in London, EMA has now moved to Amsterdam. This is the medicinal product development pathway from discovery through the non-clinical studies, and my previous speaker has referred a lot to the non-clinical studies and to the various animal models. The clinical phase, exploratory, confirmatory, then marketing authorization, and then the post-authorization studies. And I would like to show you with the case studies a little bit that the principle for ATMPs is the same, but of course we have to take into account the specificities of this problem, and then there will be deviations from the clinical development, for example, of small molecules or other biologicals. And so the principles are the same, and I will take you a little bit then through the exploratory trials and the confirmatory trials, looking at how we assess pharmacodynamics, pharmacokinetics, dose finding and safety. Starting with this contracyte-containing product, a typical tissue-engineered product on the left side, where the product comes from a patient's own biopsy from a healthy part of its knee cartilage. The cells are then expanded in culture and given back to the patient, either in the form of cell suspension or placed on a matrix to repair the knee cartilage defect. And the rationale for the use of autologous cultured chondrocytes or autologous chondrocyte implantation was actually developed by Max Brittberg from Göteborg University back in 1994, the first publications based on the rationale that the currently used methods of when a knee cartilage defect is repaired often result in a scar tissue in a not hyaline type of cartilage, and that repair by patient's own cultured chondrocytes has a higher chance to result in hyaline-like tissue that is durable. And now we come to the differences if you look at those tissue-engineered products versus other non-ATMP developments. In, for example, that conventional ADME studies are not relevant. We rather talk about cell distribution, viability, trafficking, growth, phenotype. We talk about cell biodistribution. And in the case of a chondrocyte-containing product implanted into the knee, we would not require pharmacokinetic biodistribution studies. Those would be referred to the non-clinical development. The pharmacodynamic parameter in that case would be structural repair, assessed either by histology or by MRI. We would, in this case, also not necessarily require dose finding studies. And one specific aspect for these type of tissue-engineered product is that the safety assessment includes both the product and the surgical procedure. And in this case, we would, because this is not a rare disease, in contrast to most of the other disease areas where ATMPs are developed, in this case, we would ask for a randomized controlled trial using the autologous chondrocyte implantation versus, in this case, for example, microfracture as a standard procedure. And this is what has been done for these three chondrocyte-containing products that have been authorized. Now, second case, CAR T cells. And here you see the two CAR T cells that we've authorized in 2018. Both were developments derived from the US, from academic centers, University of Pennsylvania, Sloan Kettering Cancer Center, NCI. And you see here the indications. Yescarta is authorized in non-Hodgkin's, in a subtype of non-Hodgkin's lymphoma. Kymriah, the other CAR T cell, is authorized in non-Hodgkin's lymphoma and in patients with B-cell acute lymphocytic leukemia. Both products are CD19 targeting CAR T cells. They are still manufactured in the United States, and both companies, Gilead and Novartis, are in the process of building a manufacturing site in Europe for the European patients. You can see here that, well, CAR T cells are autologous T cells genetically modified ex vivo by transduction with a retroviral vector. And they have complex and variable structures. The genetic modification results in intracellular co-stimulatory signaling domains, and you can see the different signaling domains in those, by now, more than 100 and 200 different CAR T cell developments. They vary, and they also have different extracellular antigen binding domains. The majority of currently developed CAR T cells are targeting CD19, but there are many other antigen targeting CAR T cells under development. And the mechanism of action is that once infused, they bind to antigen expressing cells. These can be tumor cells, but also normal cells. In the case of CD19, it's also the normal B cells that they bind to, and causes a certain type of toxicity. Then the co-stimulatory domain activate downstream signaling that leads to T cell proliferation and effector function and death of the antigen carrying target cell. And besides that very specific mode of action, they also have specific kinetics, and the CAR T cells are the first medicines that we call living drug. Here you see the kinetics that published by Maud from University of Philadelphia in 2014 of these CAR T cells, and you can see that from after infusion and upon meeting their target, these patients' own T lymphocytes that are genetically modified expand, and they persist over a certain period of time. And it was very surprising for those investigators to see that within a month, in many cases, the tumor or the leukemia disappeared. So they have a very specific kinetics and a specific type of action and specific toxicities as well. And you can see here that another specificity is that they are patient-derived. So the patient that is supposed to be treated with CAR T cells needs to undergo apheresis. Then the T cells are separated, they are shipped, they're frozen, shipped to the United States, to the manufacturing site, then where the manufacturing takes place, and they are frozen again, they are shipped back to Europe and administered intravenously to the patient. And before that, the patient has to go lymphodepleting chemotherapy. So it's a complex procedure, and when we assess and evaluate these type of products, we look at the whole procedure, and we have to also assess how many of these patients that were apheresed actually were able to receive the products. A lot of additional things that have to be taken into account in the evaluation of these products. And one other thing is that there are a lot of factors that influence the efficacy and the safety of these products. It's not only the product itself. These are patient-related factors, the tumor type, the pre-treatment of the patient, because the pre-treatment, again, could have an influence of the apheresis product. Then quality of the apheresis material, but also the tumor load. It has been observed that patients with a high tumor load have higher toxicity, for example. Other factors, product-related, the CAR construct, the T-cell subsets, transaction efficiency, the potency of the product, and the viability. And then others, production capacity, the lymphodepleting chemotherapy, dosing and schedule. All these are factors that influence the efficacy and the safety. So when we look at the exploratory trials, what would we require? Yes, in this case we would require pharmacokinetic and biodistribution data, PD, pharmacodynamic data. In this case, inflammatory regulatory cytokines are used to help describe the pharmacodynamics of the product. Dose finding for a living product is something that it has been found to be difficult. In the initial times, dose finding studies have been performed with CAR T cells, but they are only partially informative due to the in vivo expansion. And then the confirmatory data, ideally we would have randomized controlled trials versus standard of care. For these two products, they were where patients were included with last line treatment. There was no standard of care available, so we accepted the single arm non-controlled phase 2 study at marketing authorization. And it's typical for these products that are developed in orphan diseases that the clinical development will be composed of phase 1 and a phase 2 single trial, but sometimes also of combined phase 1-2 trials. You see here the toxicities. Main toxicity is cytokine release syndrome. It's a toxicity that is related to the mechanism of action of these products. Another important toxicity for the CD19 targeting T cells is neurotoxicity. And these toxicities have guided us in the assessment of a marketing authorization to ask for conditions, for post-marketing conditions to assure the safe use of patients treated with CAR T cells. And one of the conditions was that a monoclonal antibody that can reduce cytokine release syndrome be available at the treatment center. Another condition was that this product can only be used in treatment centers that have previously been qualified. Qualification program, a site qualification program, educational programs, of course, is also important to train educational material for physician healthcare professional, and we are asking and have asked for post-authorization studies to be conducted. Okay, that's about CAR T cells, and the third products that I wanted to guide you through are these adeno-associated viral vectors. Have seen in the last 3 years many of those in clinical development, and they are specifically suitable and have already been addressed by the previous speaker to treat monogenetic diseases. They are delivered in vivo, so this is an in vivo gene therapy. The advantage of AAV vectors is that they are not integrating, but they are episomal, and the duration of the expression of the transgene is over a prolonged period of time, 1 to 3 years, and even longer. They are stable. We have to deal with pre-existing host immunity, and there are some toxicity concerns, but in general they are quite suitable vectors to treat monogenetic diseases. And here you can see some of these more than 30 AAV products that we've seen in scientific advice and that are in clinical development. One of those products has received a marketing authorization in Europe last year, and that's Luxturna, where the therapeutic principle is that the transfer of the human RPE65 cDNA to retina cells, and this is in patients, in children that have a vision impairment due to an RPE65 mutation and a non-functional gene. So this product is not delivered intravenously, it is delivered locally to the subretinal space, and also here we have come up with post-authorization requirements to make sure that the product is used in qualified centers only, and that post-authorization safety studies are conducted. More than, as I said, 30 of these type of products are under development, and what we see is that there is one dominant indication, and that is hemophilia A and B. You may have heard of those studies. We expect the first marketing authorization submission the end of this year, and we expect that more marketing authorization for gene therapies in hemophilia will follow, and we're really very excited to see that this is an indication where a gene therapy could make a change to patients' quality of life, patients with hemophilia. More products to come in the next 2 years, about 10 ATMPs. We have 2 presently under evaluation. You see here the couple of gene therapies in red for different indications. While in the outset in 2009, 10, 11, we've seen more tissue engineering products being developed, and now it's a clear dominance of gene therapies. And that is also reflected here in the scientific advice procedures for ATMPs in the time from 2009 to date, where you see in red the increase in scientific advice procedures for gene therapies versus cell-based therapies and somatic cell therapies and tissue engineering products. And yeah, so that's already my key message to you today, that advanced therapies that we have a regulatory frame for advanced therapies that classified as gene therapies, somatic cell therapies, tissue engineering products. The Committee for Advanced Therapies at the EMA is responsible for the assessment. They are highly personalized, either autologous or also the AV vectors, for example, are highly personalized medicines because they target a specific gene and are to be indicated only for a person that has this gene mutation or defect. The majority of so far authorized ATMPs are autologous products, like the contrasite-containing products, like the CAR T cells and other products. But research and development activities for off-the-shelf gene therapies, like the recombinant AV vectors, are rapidly increasing, and these treatments offer new treatment options for patients with rare diseases and patients with unmet medical need. So in general, it's a case-by-case development and a case-by-case evaluation that has to be focused on the specificities of these advanced therapies. There is need for specific expertise, and I would say be prepared for more to come. So thank you very much.